Very good afternoon to everyone. I'm sorry about the technical glitch that happened. Uh, I think this is the problem of hi-fi gadgets. Uh, I'm sure all of you are aware of lymphatic filariasis. Uh, my talk will be today on diagnosis and xeno-monitoring for lymphatic filariasis. Can you go? So, as you all know, Dr. Hariharan, can you remove that viewing ICMR NI application?
I think you're watching only PPT only, then, sir.
Yeah, there is one white box coming.
Okay,
Viewing
okay,
ICMR NI's application. It covers the titles.
okay. Is it okay, sir? No.
No, still it is there. Okay, anyway.
Okay,
So
but
you
it's
know
not
the
showing.
clinical spectrum of lymphatic filariasis. It causes subacute asymptomatic condition, which is lymphatic due to lymphatic abnormalities such as dilatation of and lymphangitis. It also can cause renal abnormalities and other minor clinical symptoms like proteinuria, then fever, chills, malaise, headache, etc. Then in acute condition, you get adenolemphangitis, lymphangitis and adenitis at groin and inguinal regions. And there are other minor acute symptoms also. Then the chronic conditions are hydrocele in males, and of course the lymphedema, chyluria, and other clinical symptoms. And there is the most common chronic symptom is tropical pulmonary eosinophilia, that is nocturnally paroxysmal cough. So these are some of the clinical manifestations. You all must have these disease conditions when you go to the field for surveys. So filariasis is a deformity caused by the nematode parasites, and the clinical spectrum I just now listed before you. It impairs the mobility and also causes a lot of impact on social life of the affected individual. And in younger children or adults, it can also cause problem in educational activities, employment, marriage prospects, and also marital relations and sexual life of the married youngsters. Basically, it is a poor man's disease. It affects poorest of the poor, not only in India, also in other developing countries. So it causes significant socio-economic loss to the affected individual. And in India, as per the VCRC's estimate, in India alone suffers an annual economic loss of about 1 billion US dollars. You can see how much financially our country will be losing because of lymphatic filariasis. So considering this as a major public health problem, the WHO declared it as disease for elimination. And in India, we targeted this for 2015, and since we could not achieve, we switched over to 2020. Now, as per the WHO strategy, now the elimination deadline is by 2030. So this is the global picture. You can see the number of cases in different regions of WHO, and as you can see, CRO contributes the major burden of lymphatic filariasis in the whole world. So in India, as you all know, you have been very familiar with the filariasis problem in India. So there are 553 million people are at the risk of filarial infection, And already those who are affected with the deformities, that is lymphedema and hydrocele, there are about 21 million cases. And as of 2000, there were 27 million parasite carriers, mainly the MF carriers, asymptomatic cases. But now, over 15 years, 15, 16 years of elimination program in India, especially the MDA with the combination drug, the number has been reduced much more, but there are still many districts which are persistently reporting the microfilaria prevalence more than 1%. So this is the statistics. This is a bit old. Globally, about 1 billion people are in the risk of filariasis. In India, there are 28 states, 7 union territories, and 17 states are endemic, and so also 6 union territories are endemic for filariasis. Total number of out of 466 districts surveyed, 257 districts were found to be endemic about a decade ago, but now this number has been increased because of addition of new districts which were recently found to be endemic for filariasis. So this is the picture, the burden of LF in global scenario as well as in India. So there are few drugs currently available. So the diethyl carbamazine is the one which has been in use for almost 60 years. More recently, albendazole was added to the program, and just this year, last year, a few districts, 13 districts, as you know, in 13 days, even the third drug is added on an experimental basis. Ivermectin is supposed to reduce the MF density drastically. And in Karnataka, Yadgir districts and Sindega in Orissa, Jharkhand, the trial is going on for the last few years. And there seems to be quite a good picture about the outcome. So basically, filariasis is caused by three parasites, as you all know: Wuchereria bancrofti, Brugia malayi, and Brugia timori. Of course, in our country, the major parasite that causes filariasis is Wuchereria bancrofti. It contributes to more than 90% in our country. A minor proportion of the cases are due to B. malayi, which has been drastically reduced over the years, and it is now prevalent in a few small pockets of Kerala and four more states, especially Orissa. And Wuchereria bancrofti is transmitted by Culex quinquefasciatus. So as you can see, the problem of filariasis, psychosocial impact of filariasis. So this lady is the only bread earner to the family, but you can see that her legs are deformed. I don't know how far she will be able to function as a tailor. Next. So it causes great social burden to the affected individual. It causes stigma, anxiety, ostracization for family and community. So it leads to psychological trauma, and in adults causes sexual dysfunction as well. And it impairs mobility, travel, educational opportunities, and employment opportunities, even the marriage prospects. Next. So, considering this as a major public health problem, as I said, there was a declaration by World Health Assembly that it should be targeted for elimination, and the major strategies for elimination are mass drug animal single dose of combination of DEC and albendazole for the community, and more recently, ivermectin is the addition. And the other arm of the elimination program is morbidity management. So which basically focuses on leg hygiene. Next. So this is the life cycle of filariasis parasite. You all are very familiar with it because you have been working in this line for quite some time. But when it comes to diagnosis, there are three stages which are targeted for diagnosis. One is microfilaria in humans, so which can be done by microscopy, and that is the diagnostic part of this lecture. And xenomonitoring. The xenomonitoring is a process by which the parasite is detected in the vector mosquitoes, whether it is Wuchereria bancrofti or B. malayi. These two parasites are detected in mosquitoes by dissecting the mosquitoes, wherein you may find microfilaria recently ingested, which molds into L1 larvae and then to L2 larvae and L3 larval stage. L3 larval stage is the infective stage of the parasite, which is transmitted to a new host by the bite of the mosquito, a female mosquito. And this is very important parameter because you can assess the transmission by dissecting the mosquitoes. And especially the L3 stage, which is transmitted by the bite of mosquito to new host, gives us infectivity rate, whereas if you find other stages such as MF or L1 or L2 or L3, that gives us infection rate in mosquitoes. So this is a very important epidemiological parameter for assessing the transmission of LF in an endemic area. You can also quantify the number of parasites in the humans by looking at the blood slide collected during the night time for the nocturnally periodic strain of the parasite. So take a drop of blood and examine under the microscope, which you people have been doing for quite some time. So let us see what are the things that are required in the process of elimination of filariasis. In the initial stage, you will have to map the filariasis in different areas, endemic areas, or even in non-endemic areas if it is there. So the tools that you can use for mapping, the mapping and stratification are: one is the conventional microscopy, wherein the blood collected during the night is examined under microscope, or you can collect daytime blood sample and subject them to immunoassays such as antibody assay or antigen assays. Currently in the market there are two commercial tests. One is, earlier it was ICT, now it is a strip test, FTS, or you can also do OG4C3 ELISA, which is of course a little difficult because you need ELISA reader and other laboratory based equipments. But for the strip test, you don't need to have any machinery, and it can be done at the site of the either hospital or in field. So this is very handy and useful technique wherein you are detecting the antigen by using a strip test. Then once you stratify, then you will have to implement the mass drug administration. And during the process of 5 to 6 years, as per the WHO strategy, you will have to monitor the infection both in vectors and in humans. In vectors, you will be doing it by xenomonitoring, and in humans, you will be doing it by checking the antigenemia or the micro. And finally, in the certification process also, you will have to use both the diagnostic techniques, either the antigen or antibody in humans, or microfilaria in humans, or the xenomonitoring for assessing the infection in vectors. So this is the vector of Wuchereria bancrofti in our country, Culex fasciatus. You can see the tummy of the mosquito is fully fed with the blood of the human host. So these are the kind of breeding places where the Culex fasciatus breeds. It's a dirty water mosquito. It breeds in drains, puddles, pools of dirty water, or even in unused wells where, or disused wells where people tend to throw, if the well is not being used, people tend to throw garbage and other things, and that becomes a heavenly place for Culex fasciatus to breed. So this is the vector of Brugia malayi, Mansonia species. Mansonia endulifera is the major vector, and it breeds in clean waters like this, a waterway in Kerala. You can see the ponds, and you can see the ponds around the house also. So this species of larvae of this mosquito breeds always in a water body where you have the water weeds such as as you can see here. So it attaches to the roots of these aquatic plants for respiration. So which means that unless you have these water weeds, Pistia or Eichhornia, Mansonia will not be able to breed. So if you can remove these weeds, so you are controlling Mansonia mosquitoes and Drugia malayi. That is about something about the filariasis and the disease and the vectors. And now let us see what are the diagnostic options available for us. In humans, you can also diagnose by way of symptoms. If there is a, pulmonary eosinophilia, or there is a swelling in the leg, distal parts of the legs or hands, or the scrotum. So those are the symptomatic diagnosis. And even if you can give the drugs and if you see no more symptoms, so that is the way of therapeutic diagnosis. And as far as parasitological diagnosis is concerned, as I said, you have to collect a drop of blood from the individual suspected to be affected with the filariasis, either daytime or nighttime. In India, we have nocturnally periodic strain of Chirarea bancrofti in the mainland, and in Andaman Nicobar Islands, you also have uh, diurnally periodic strain of the parasite, which means that you can even collect blood sample during daytime. So you can make a thick smear, or you can also do by knots concentration or membrane filtration. And if you feel it is very difficult to collect blood sample during night time, you can give a dose of DEC, whereby the parasite appears during the daytime. So these are all the parasitological methods that are available. And but the most user-friendly method is the uh, the drop of blood collected during the night time, and followed by you will have to smear on the blood slide, and you have to stain it with JSB, and then do the microscopy and look for the presence of microfilaria. Then what are all the immunological techniques that are available? In early days of immunodiagnostic development for filariasis, there were agglutination tests, then indirect hemagglutination test, followed by antibody assays. But these were not sensitive or specific, especially antibody assays were blamed because they can only detect either the past infection or a recent infection, not the present infection. So therefore, these three types of diagnostics were found to be of no use. Then came the OG43C3 ELISA, which was quite good, highly sensitive, and we tested several thousand samples by ELISA for our epidemiological studies, and we found that it was highly specific and sensitive, but it needs laboratory setup. You need ELISA reader, washer, and other associated equipment, refrigerators, and so on. So that was possible only if you have a centralized laboratory, if you want to use the ELISA, enzyme-linked immunosorbent assay. But the ELISA was superseded by uh, field-based diagnostics, or rapid diagnostics, what presently they are called as. And one such technique was immunochromatography test, which detects the antigen of filarial parasite during daytime. But this was earlier supplied by the Australian company. Now that company is no more selling it. Then came Alair, and now you have FTS, filarial strip test. So which is currently being used, little less expensive, So this technique is very handy, and currently it is being used in elimination program for monitoring the elimination program, as well as for TAS, transmission assessment surveys, and also in the post elimination surveillance stages. So in the three use cases, this test is currently being used. Then you also have some molecular biology based assays. The earliest one was hybridization, DNA hybridization, which was very cumbersome and less sensitive, but that was uh, superseded by PCR assays. PCR assays. Today we have conventional PCR, then the real-time PCR. Of course, some people do the sequencing for identifying the genotypes of the parasite. So real-time PCR and regular PCR, polymerase chain reactions, are very, very highly sensitive and specific. But the problem when you want to diagnose the blood sample, this is little difficult because the DNA of the parasite is not there in the blood. Unless there is a parasite, at least one parasite, you will not be able to get the results. So, but PCR has been very important for xenomonitoring. Next. So this is the picture of microfilaria in a blood smear. So you can see the nice curled parasite here. And in Brugia malayi, how to differentiate between Wuchereria and Brugia is that in the tail end you have additional prominent nuclei in the tail end of the Brugia malayi microfilaria. So by the presence of the nuclei present in the tail end, you can differentiate between Brugia malayi and Wuchereria bancrofti. So these are the two assays, immunological assays. As I said, OG4C3 is an ELISA technique. Uh, this is highly sensitive and quantitative. You can estimate the concentration of antigen present in the blood by OG4C3 assay, which is normally not required for elimination program as long as you do qualitative test, presence or absence. That's good enough for the elimination program. This is highly sensitive and highly specific, which we tested in our laboratory almost about 15 years ago. But the problem with this assay is that you need to have a lab setup. Unlike that, the immunochromatographic card test is also highly sensitive and specific. Added advantage is that this is a point of care diagnostic. You can use it at the site of the patient or the field. So these techniques detect the infection during daytime, and they are highly sensitive and specific. And especially the ICT card or the FTS, which is being currently used, is very simple, rapid. In 10 minutes, you can detect the infection in an individual. So as of now, OG43C3 is only used for research purpose. So if you want to diagnose the lymphedema cases, whether it is due to filariasis or other reasons, so you can go for ultrasound studies. So this is the clinical diagnosis. So this is another technique, lymphoscintigraphy. These are only for checking the lymphedema where the you can find out where the blockage of lymph is taking place. But these are not for routine diagnosis. Now they have remained as tools for research. Now this is again the lymphoscintigraphy, different pictures of legs, knees, and thighs and pelvis. So much so about the diagnostic techniques to detect the infection in humans. So if you have any questions, we'll discuss once I finish the xenomonitoring part of it. So xenomonitoring is basically a technique of detecting the parasite in vectors, whether it is microfilaria stage or L1 stage or L2 stage or L3 stage. So these findings will be very useful for assessing or determining the infectivity rate and infection rate. Infectivity rate is the proportion of parasites which are in infective stage, that is the L3 stage. Infection rate is calculated by taking all the stages into account. So that is the difference between infection rate and infectivity rate. But the problem with the dissection and microscopy, which you saw in the earlier stage, is that it has been very useful for the past 50 years to assess epidemiological situation in an area. In the sense, it has a limitation of number of samples that can be subjected to xenomonitoring in a day by a group of staff. We have worked out at VCRC if 5 people are engaged in dissecting the mosquitoes and dissecting them and looking for parasites. At the best, they can assess infection in about 100 mosquitoes. So, which goes to say that it's highly cumbersome, and also it needs a good skill of dissection and identifying the parasites in a large debris of the dissected mosquito. So, if there are people who are very casual, they may just mistake it with the tissues of mosquito. So there is a sub level of there is a problem of uh, subjectivity. So it all depends on the skill and interest of the technician who is dissecting the mosquitoes and detecting the parasite in the mosquito debris. So that is one problem. The other problem is that in a program like lymphatic filariasis elimination, and in a country like India, you will have to dissect millions and millions of mosquitoes to assess the transmission. So, in such a case, the dissection microscopy technique will not be very suitable. So, therefore, there was a need to develop alternative techniques for rapid and large scale detection of the parasite in vectors, that is, for xenomonitoring. So the molecular biological techniques developed about 30 years ago came very handy for xenomonitoring, and one such development was the polymerase chain reaction, which holds great promise as a diagnostic tool for a variety of pathogens. So we have developed a very simple xenomonitoring technique. So the technique involves no very expensive kits or reagents. So all one has to do is to collect mosquitoes, vectors, separate the female mosquitoes because they are the ones which will be harboring the parasites. Dry a pool of mosquitoes at 95 degrees centigrade for 3 hours. Grind them to a fine powder in a tube. Add 100 microliter of tea buffer, vertex briefly, and boil for 10 minutes. Centrifuge for 10 minutes and take the supernatant directly into the PCR tube. So when you do the polymerase chain reaction and run the amplicons, the amplified products on a gel, you will see a band of 188 base pairs. So that shows that the mosquito had the parasite, filarial parasite. So we extensively tested this technique. So unlike the techniques used elsewhere in Europe and America, where they use the Qiagen and other kits, which cost something like $10, in the sense the sample processing alone will take up 7 to 800 rupees. But our technique, which is based on laboratory buffer, commonly used laboratory buffer such as TE buffer, it's very, very inexpensive. Probably per sample, the cost may be about 1 rupee. So compared to the cost of the commercial kits and our technique, there is a lot of savings on the money that is used for extracting the DNA. Nevertheless, this DNA is very, very useful for PCR assays. We compared the technique in field and also in the laboratory. So it is as sensitive as the technique that uses the commercial kits. So we trained people from different ICMR centers in this technique, and most of the people who participated in the workshop did not know the molecular biology techniques. They didn't have any experience in PCR assays, but still, as you can see, they were with a one day's training, they were able to do this PCR very easily. We conducted this workshop in as early as 2003. As you can see here, all of the participants from different institutes, although they didn't have any molecular biology experience or experience with PCR, they were all able to get the same results. The only person who was rather very good molecular biologist was Dr. Triveni Krishnan from NICET, and all others were able to get the same results as good as that of Dr. Triveni's. So that shows that this technique is very, very user friendly, and anybody with a training of about a week can perform this PCR without any much of a problem. So you may ask whether the technique PCR is as good as the conventional techniques of microscopy or antigenemia. We conducted large field trials and we compared the sensitivity and the relationship. We worked out the relationship between microfilariae and PCR, antigenemia and PCR. They all gave the same kind of relationship, which means to say that PCR can be an alternative technique for transmission assessment surveys. So that is the PCR, conventional PCR, which gives us a handle to assess the infection rate, sorry, because it detects all the stages of the parasite in the vector. But actually, you would also need to know whether there are L3 stages in the vectors. So we did lot of genomic work. We did subtractive hybridization of the genes of different stages of the parasite, and we identified two genes which tells us whether the mosquito has infective stage, that is the L3 stage of mosquito or not. And if that is the case, we'll be able to work out the infective rate also. So we have identified two genes which can be used in the qPCR as well to determine the infectivity rate by way of xeno-monitoring. Then there is another question. We are administering different drugs, diethyl carbamazine and albendazole. So far, there is no report of resistance against DEC by the parasite. But albendazole is definitely a target for drug resistance by the parasites. In veterinary field, when they used albendazole, many of the parasites very quickly developed the resistance to this particular drug. So we developed a method of detecting the resistance against the albendazole based on it is based on a beta tubulin gene, because beta tubulin is the target for albendazole. So we were able to sequence this gene from different parts of the country, like Pondicherry and other places. You can see 10 different places we collected the parasites and did this assay, and we found that still there is no development of resistance to albendazole in these areas. And subsequently we developed a simple PCR assay. So now people can detect the resistance if there is any to albendazole by using this methodology. So these are all the things which I had to share with you, and these are my students and staff members who helped in developing these diagnostic techniques and xeno-monitoring techniques. So I am thankful to most of my people in the lab, and also I am thanking the NIE colleagues and all the NBBDCP colleagues for giving me this opportunity to share my views and the techniques which we have developed in our laboratory. I would very welcome the questions from all of you. Thank you. Thank you very much.